Good afternoon, everyone. My name is Klose. I'm going to present this webinar today with the title Why Population Health Studies Must Run Lipidomics. So let's get started. The first population health or cohort study, which led really to conclusive insights into associations between lifestyle habits and disease and mortality risk, was the British doctor study. It was started in 1951, and it was a study among British doctors, as the title suggests, and provided convincing statistical proof that tobacco smoking increases the risk of lung cancer. And here are the data. So the study clearly showed that smokers die earlier than non-smokers, and that quitting smoke is beneficial to your health as it extends your lifespan, as you can see here in this survival curve. So in summary, the study really provided conclusive evidence of a linkage between smoking and smoking and lung cancer, myocardial infarction, respiratory disease, and other non-smoking and other smoking-related illnesses. So in total, smoking decreases lifespan up to 10 years. And the earlier you stop smoking, the better. That was the outcome of this particular study. Another famous cohort study is the Framingham Heart Study. It was started in 1949 and led to the Framingham Risk Score, which is a well-known estimate for the 10-year cardiovascular risk of an individual to obtain a cardiovascular disease. So the Framingham Risk Score is based on age, sex, smoking, blood pressure, and the status of lipid metabolism as assessed by blood levels of high-density lipoprotein and cholesterol. And this really highlights also the importance of lipids and lipid metabolism in disease etiology. And lipids do not only receive lots of attention in the context of CVD, but also are an important aspect in many other diseases, as can also be seen from the publication record in PubMed using the term lipidomics. So as you can see here, since the early 2000s, the numbers of publications using the term lipidomics has increased dramatically, with now about 2,000 publications per year. Lipids are a structurally very heterogeneous group of molecules. As you can see here in this slide, they consist of an apolar part, this end here, usually made up of fatty acids, and also a polar part, which is called the lipid head group, shown here. For example, glycerophospholipids. For them, we can achieve a great variety of structural diversity by having different fatty acid attached to the glycerol backbone, shown here. And these fatty acids differ by, for example, number of carbon atoms in the chain and also numbers of double bonds in this fatty acid chain. They also differ in the way how they are linked to the fatty acid, to the glycerol backbone. For example, here we see an ester bond, while here we have an ether linkage, which has like tremendously different physicochemical properties. In addition to those heterogeneities conferred by these fatty acids, we have also different of these polar head groups, which I talked earlier about. And these head groups give rise to these common lipid classes that many are familiar with, like phosphatidylcholine, phosphatidylinositol, but there can also be sugar head groups attached here, giving rise to glycolipids, for example. And in this way, thousands of different lipid molecules can be produced by our bodies. In theory, it's even tens of thousands, but in practice, we see a few thousand or so in a typical biological sample. And where do we find those lipids? You are probably all familiar with lipoproteins. So as you know, lipids are nutrients in our diet. And once we have eaten them, they enter our bloodstream in structures like those lipoproteins. And these lipoproteins consist of an outer shell, shown here in blue. And this outer shell is mostly made up of phospholipids, but also lipoproteins and cholesterol molecules. And in the core of those lipoproteins, we find neutral storage lipids like triglycerides and cholesterol esters. And these lipoproteins are actually the vehicles that are used by our bodies to transport lipids to the various organs, where they serve as nutrients or building blocks for cellular structures in the form of membranes. And those membranes look basically like this. So this is a typical cellular membrane as we find them surrounding our cells or forming intracellular structures such as organelles like Golgi apparatus, endoplasmatic reticulum, and so on. But not only do these lipids form the surroundings of the cells, but they also are the medium in which transmembrane proteins are actually embedded. And it's important to know that about 30% of the entire proteome of a typical cell are transmembrane protein, and many of them play important roles in cell-cell interactions, signaling process that are fundamental to disease mechanisms like, for example, diabetes or cancer. And these lipoproteins or these proteins in general in the plasma membrane or in any other membrane basically interact with the lipids, but they're also influenced by the lipids. So it's really essential to understand lipid composition of a membrane in order to understand how proteins, transmembrane proteins, peripheral membrane proteins actually are functioning. The way lipids are approached analytically has changed quite a bit in the past years. So on the left-hand side, you see a typical thin layer chromatography picture or photograph. And with this methodology in which you basically are able to separate lipids in a given lipid extract based on their retention behavior on a silica plate, it is possible to basically identify lipids on the level of lipid classes, so based on their head group structure, essentially. And this was the preeminent methodology in the 80s and before. Most of lipid analysis was done based on thin layer chromatography, and the lipids were visualized based on the lipid classes simply by staining those plates and getting these bands that look very similar to protein gels or DNA gels. In the 90s, mass spectrometry became more and more available in biological laboratories. And it was then possible to not only quantify and analyze lipids at the level of lipid classes, but really start looking into the molecular composition, lipid composition of samples. And in the 90s, we speak mostly about quite simple mass spectrometers, such as triple quadrupoles, which allowed, with their low resolution, they allowed the analysis of lipids on the some compositional level. While later on in the 2000s and onwards, more and more high resolution mass spectrometers became available also to biological labs. And since then, it was really possible to obtain detailed structural information about lipids in a complex biological sample by also fragmenting those and obtaining fatty acid information, etc. So there was really quite a strong development in terms of mass spectrometry and in general analytical capabilities that really enabled lipidomics, which then really resulted in the steep increase of numbers of publications applying the technology, as I've shown in an earlier slide. At Lipotype, we have established a shotgun lipidomics technology for the quantitative analysis of lipids that basically comprises two essential parts. So the first part is the lipid analysis itself that takes place in the lab. And the second part is the data analysis part, which is equally important. So to talk a bit about the lipid analysis in the wet lab, basically how it is actually done. So we start off with a biological sample, which can be a blood sample, a tissue sample, or a cell culture sample. And we extract the lipids out of these samples by the use of organic solvents, such as chloroform and methanol. During the extraction, we also add standard molecules, standard lipids to the sample in order to be able to do a quantitative analysis. So the standards we put into the sample are of known quantities, And based on this information, based on these quantitative standards, we are able to actually quantify the amounts of lipids in the biological sample. The sample extraction itself is done with a robot. Everything is done in a 96-well format, and the extracts are then taken and directly infused into the mass spectrometer. For the sample infusion, we use a so-called Hamilton Adveon nanomate ion source, which is also working in 96-well format, and basically picks the sample and sprays it into the mass spectrometer. And then for mass spectrometry, we use thermo-Q exectives, which are based on Orbitrap technology, meaning they can provide really, really high mass resolution and acquire a mass spectrum of a complex biological sample, such as a blood sample, in less than 5 minutes, and record the spectra. And once the spectra are recorded, they are not very informative for a layperson. So what we have to do is we have to annotate those spectra and determine actually which lipids are present in the sample. And that is done in the data analysis part. And for this lipid identification, for example, we have developed a proprietary software called Lipotype Explorer, which exactly does the annotation of the peaks in the mass spectrum to give us the information which lipids are actually present in our sample. And once we have identified those lipids, we can also identify those internal standards that we have added before the extraction, do all kinds of normalizations and visualizations of the data. And for that, we can, for example, use a software tool, which is called Lipotype Zoom or Lipotype Zoom Pro, which allows to visualize data, do sophisticated statistical analysis in order to be able to interpret the results of the lipidomic study. In addition to our shotgun lipidomics platform, we have recently also established a targeted LCMS based platform, which is based on chromatography and triple quad mass spectrometry. It has the advantage that it is more sensitive than the shotgun lipidomics approach. So when we really want to analyze lipids that are of very low abundance in particular samples, this is the technology platform we would go for. It has also increased specificity. So because of the chromatography, we can separate lipids from each other that have similar mass. And in this way, we can really distinguish them from each other and can do, as for the shotgun approach, by using internal standards, do a quantitative analysis here. And usually, again, also this platform is based on a classic folic extraction, for example, using organic solvents. And most of the times, we're using reverse phase chromatography to separate lipids from each other. Okay, so much to the technology that we are applying in order to do lipidomics itself. Another important aspect is quality control. So as we are really talking today about large-scale projects with hundreds and thousands of samples in the context of cohort studies, this data quality and quality control in general is key. And essentially, data quality is based on highest analytical performance. So we have to make sure that when we analyze thousands of samples, that the equipment is always working optimally, that everything we do in the lab is done correctly. And in order to make sure that this is really the case, in addition to the whole panel of system suitability tests that are run prior to actual sample analysis, we assess the method of performance based on reference samples and blank samples that are run along the actual study samples. And I'd like to show you briefly how we do that. So here is a schematic of the 96-well plate design as we usually use it in large-scale screening. So here marked with the green R are the wells in which we would put reference samples. These can be pooled plasma samples or any other pooled tissue material that is appropriate for a given study. And the red B here indicates a blank sample. That is a sample that is treated like all the other samples in terms of internal standards added, extraction solvents used, etc., but it does not contain an actual sample. So it's really an empty extraction without any biological material. So this helps us to make sure that, so first of all, it allows us to track, using these reference samples, it allows us to track reproducibility of the method, and the blank samples in between here allow us to make sure that there's no cross-contamination from one well to the other when analyzing one plate in a row. So, and with this particular plate design, we are able to monitor parameters such as analytical drift and potential batch effect. And what that is, I will talk about in the next slides. So, analytical drift. Basically, analytical drift is the stability of a signal over time in a given analytical run. So, for example, when we analyze a full 96-well plate, it takes about 8 to 10 hours, depending on the sample type, and these samples are analyzed sequentially. So, from position 1 to position 96, shown here on the x-axis of this plot. And for the reference samples, which were like arranged on the plate in a way similar to what I've shown in the previous slide, we would expect that with a stable analytical technology, we would expect consistent results for a given lipid in those reference samples. So basically, as shown here, we have a straight line here, which is basically a linear regression of the amounts of the lipid class of lysophosphatidyl-choline over the course of a run analyzing a 96-well plate. So this is really what we want. If we now had seen like a drift of the signal, like upwards or downwards, we could detect it with this plot, and we could also, based on this linear regression, also correct for these potential drifts in case it's not too heavy. So that's one important aspect for why we are using those reference samples. Another important aspect is controlling so-called batch effects. So when we are looking at thousands of samples, we will have to run those samples again in 96-well format, meaning that we analyze them on many different days in a row, basically, or even like in different batches, really weeks apart. And what we have to make sure is that in each of the runs, we get the same results for our reference samples, so these data can actually be compared with each other. And what is shown here is, for example, the amounts of triglycerides on different well plates, which were run on different days, maybe even on different mass spectrometers. And with this plot, we can make sure that the data are actually comparable and reproducible from day to day, from machine to machine, sometimes from operator to operator. And in this way, we can really show that the analytics is robust, and we can really produce reproducible data also in large-scale projects. Taking all this together, we can achieve a pretty good analytical performance of our shotgun lipidomics technology. So the technical variation, as assessed by looking at the reference samples I mentioned earlier, is typically below 10% coefficient of variation. We cover a wide dynamic range, which is typically larger than 4 orders of magnitude, which means we can measure lipids in the range of about 1 picomole to the range of about a few thousand picomoles. So they're all within a dynamic range where we get a linear response and can basically, therefore, very nicely do a quantitative analysis over this large concentration range. The methodology is also pretty sensitive. Usually we are speaking about a nanomolar range sensitivity, which boils down to 0.001 mole percent of total lipid content, which is, for a direct infusion approach, a really, really good value. And this we can all achieve from very low sample amounts. For example, 1 microliter of blood plasma is sufficient to get a full profile. Talking about tissue material, a few hundred micrograms of tissue are sufficient. And since it is a direct infusion approach, taking a few minutes only per sample, with our current setup, we can analyze about 400 samples per day, which is key to really analyze large cohort studies as we have done in the past. In addition to the analytical validation, the biological validation or understanding of the biological variation of a given sample type is almost equally important, I would say. And therefore, we usually want to understand factors that influence the lipid composition of a given type of sample, be it a certain tissue type, be it the lipidome of a certain organism, or in that case that I'm going to talk about in the next few slides, about a healthy human blood lipidome, how it is affected by factors such as age and sex, and what are the consequences and the conclusions. So, and in that study that we did together with our colleagues from the Finnish Institute of Molecular Medicine in Helsinki, we have conducted a baseline characterization of human blood plasma, of the human blood plasma lipidome. And the study consisted of more than 7,000 participants from the so-called gene risk cohort, and we used another cohort consisting of the subcohorts of FinRisk and UFAM, with about 2,000 participants as a validation cohort. And the results that we obtained look like this. So first we look at the traditional clinical lipid parameters. So what you see here is the concentration of triglycerides, total cholesterol, HDL cholesterol, and LDL cholesterol against the age of the study participants, and color-coded by males in blue and females in red. And there we see quite interesting behavior. So for example, for the triglycerides, we see that the values at the starting point at the age of about 45 to 50 are quite different for women and men. But then with age, they start to converge. So the triglyceride amount in blood of older men goes down, while for women it increases. And we see like similar changes in different directions for all the other parameters. So what is clear here is that the classical clinical lipid parameters are dependent on age and sex of the patient or the subject in general. So, and we were interested in the question if the lipidome behaves in a similar way. Is the lipidome also, like lipidomic composition on the molecular level, also dependent on the age and the sex of the study participants? And here you see a volcano plot showing the differences between men and women across the entire lipidome. And for example, what you can see here is that men have, here all these pink circles, men have a higher concentration of triglycerides, while women have an increased concentration of sphingomyelins, here in purple, in their blood lipidomes. So that basically the result for the triglycerides confirms the result that we've seen earlier for the clinical triglyceride values, which looks at overall triglyceride content, while we look here at individual triglyceride molecules. And sphingomyelins are a class of sphingolipids that are like have very essential functions in our bodies. So the effect sizes and p-values for this plot were obtained by linear regression, and they were corrected also for body mass index and other parameters such as diabetes, cardiac diseases, lipid-lowering medication, and so on. So what we clearly see here is there is definitely a sex difference between men and women in the blood lipidome. We also wanted to understand whether these like differences between men and women also change with age, as we have seen it for the clinical parameters. And we observed again very different trends. Again, there is this one case where we had lipids that actually are very comparable in their concentration at the onset of the study, like at age 45 or so, but then they start to diverge. So there are opposite trends in the development of the concentration with age, and at the end, the concentration is quite different between men and women. Then we have cases like this triglyceride molecule where they actually converge with age. They're starting off at different concentrations, in the end, they're quite similar. We have another scenario in which the concentration, like for example, for the ceramide, stays very constant in the man, but it is increasing in the women to converge again with the concentrations we see in the man. And then we have those cases, like for example, for this phosphatidylcholine, where we have similar trends for both men and women. And here we see an upward trend for this with age for this phosphatidylcholine molecule. And basically similar things and changes we have seen across all lipid classes as shown in this complicated picture here. But I would like to just highlight the most striking findings, and that is again the diacylglycerol, triacylglycerol differences. They narrow down with age and finally result in the similar triglyceride and diacylglyceride profiles in men and women in older age. For sphingomyelin, as shown here, those differences get stronger, yeah. And for cholesterol esters, which I am not highlighting, the differences actually get bigger, yeah. And so therefore, one important conclusion from this study is that we really have to consider gender and age as confounders in these cohort studies. So we really have to take them into consideration. We would even go so far and conclude that it's not really even sufficient to have them as confounders, but to really build predictive models for like women and men separately, because they are so different and they're reacting so differently in different directions. Considering the age of the subjects, it really justifies to build separate models for men and women when talking about lipidomics-based predictive models for health and disease. And this is really now the transition to the actual use cases that I want to talk about. How lipidomics is used in cohort studies to understand how lipids reflect health and disease in the human population. And I will start with talking about metabolic disease, which is very often manifested as obesity. And you will probably all know that obesity is usually measured as body mass index, which puts the body weight into relation to body weight. And people with a BMI larger than 25 and below 30 are usually considered overweight. And if it goes above BMI of 30, they are considered obese. Above 35, severely obese. Above 40 or higher, they are morbidly obese. And obesity is a global problem, as you can see also from this slide here. In the United States, for example, there is a steady increase since the 80s in the fraction of the population which is actually obese, and currently about 40% of the US population is obese, and this really has started to develop as a big problem in the 80s, as you can see from this plot. So the fraction of people that are severely obese, both men and women, increased dramatically in the United States starting from the 80s, and we have a similar trend in most countries, also in Korea, as you can see here from this slide, which shows like the development for both men and women, but also the total for the past 10 years. So it's an increasing problem, and that's certainly not a good thing, because obesity is a risk factor for many other serious diseases. So it really comes with serious health problems. So obesity is really a risk factor for myocardial infarction, fatty liver, cancer, but also type 2 diabetes. And the costs that this brings about is really, like, for example, for the United States economy, it's causing like $200 billion of extra health costs annually, plus additional $150 billion of lost productivity due to people being not able to go to work because of sickness and so on. So this is really, it's not just a health issue, but it's also an economic burden for every country. So what we wanted to find out in one of the studies that we did a few years ago was, is the plasma lipidome actually reflecting metabolic changes that are caused by overweight or obesity? And to understand that, we did a collaboration with the University of Lund in Sweden, and again the University of Helsinki, and we used samples both from the FINRISK study and the Malmö, which is the Swedish study, the Malmö Diet and Cancer Study, and we analyzed a total of 1,250 blood samples here. Here are like the data of the population, so it's a pretty average European, I would say, population, with an age distribution around the early 50s, or for the Swedish population of the late 50s. But other than that, it's quite a typical populational study. And what we really did was we used the lipidomics data that we have obtained from these more than 1,000 samples, and used machine learning to build a mathematical model for the prediction of different obesity measures. And how that works is as follows. So we take basically the FINRISK study, which is our training data set, and build a mathematical model that basically establishes a correlation between the lipid composition of a blood sample with its hundreds of different lipid parameters. And based on this model, we see how well we can predict actually these different obesity measures. We can then pick the best model, calculate a training error, and then also use the best model and test it against a subset of the samples that we have looked at, and test whether it also works for this subset of samples that has not been actually used to build the model. And with that, we get a test error. And when we then see, okay, the model is working well, even on the test sample set, we can use it on an independent cohort in order to really validate it on data that were measured at a different time point, on subjects that were from an entirely different country, and samples collected really by also a different like clinic or whatever institution was involved in conducting the study. And by doing all this, we were able to come up with a very robust model that quite nicely helps us predict the obesity status of a person based on the lipid composition. And what you see here actually is a plot of the error of different models for example, body fat percentage, BFP, weight hip ratio, waist circumference, and body mass index. And it turned out that actually the blood lipidome works best for predicting body fat percentage. The model had the lowest error during cross-validation, and the second best model was the one for waist-hip ratio. So it seems that the lipidomics data best reflect obesity as assessed by body fat percentage. And when we go through the whole panel of model testing and validation, I just showed this for completeness to convince you that this model is very robust. So the cross-validation error, the training error, and the test error, all obtained in the FINRISK study, are quite stable. So there isn't really a dramatic increase in the error with all these different levels of testing. And when we then even go to the Malmö cohort, which we use as the validation cohort because it's entirely independent, we get a very similar error as compared with all the test errors, which is a very good sign, showing us that the model is really robust and functioning not only on the cohort that it was built with, but actually also working for a cohort that is entirely independent. And that is always a sign for a very, very good and robust model. It actually has turned out that other groups that published similar results after our study have obtained similar data. So it's not only us seeing this very, very strong correlation between blood lipidome and obesity. It's at least two more groups that see very similar trends in very different cohorts. So this is really a very solid signature for obesity in the human blood. And what you see here is a plot of the lipids belonging to the different lipid classes. And on the y-axis, we have the correlation coefficient with obesity, basically. And what you see here really is a plot of those lipids, and these are the ones that are in dark blue or dark red, that actually contribute to the predictive model. And the take-home message from this picture basically is that lipids from the entire lipidome actually play a role in predicting obesity. Just highlighting the complexity of lipid metabolism, and also highlighting that just looking at triglycerides or cholesterol is probably not the entire picture of how lipid metabolism is affected by obesity. You also see here that there are quite some differences between men in blue and women in red. So again, here this calls for a dedicated model for both men and women. In that particular case, however, we have used the gender as a confounding factor, which gave the results that I talked earlier about. So lipidomics is interesting because you can go down very low in structural detail, but you can also look at a very high structural level, meaning that, for example, as you can see in this picture here, you see here GPL in blue. This is all glycerophospholipids. Glycerophospholipids contain, for example, phosphatidylcholine, lysophosphatidylethanolamine, and so on. And each of those lipid classes, like phosphatidylcholine, then consists of many different lipid species, shown here in orange. And each species actually consists of several subspecies. That's how we call it, here shown in red. So, and the subspecies really contain the information of the actual fatty acid composition of a given molecule. And we can we can apply this hierarchy to all parts of the lipidome. And when we are trying to see what level of information, of structural information we actually require in order to build a solid model, we get the following picture. So what you can see here is basically again on the y-axis the error of a prediction model, and on the x-axis we get different data inputs for the different models. But that is not really what matters here. So the x-axis is not so interesting. What matters mostly, and the point I want to make here, is based on the color coding that you can see here on the right-hand side. So if you use a model that solely uses clinical parameters like age and sex and triglycerides, etc., we get an R square is another indicator for model performance that is slightly above 0, so not very good. If we then add lipid information on the level of lipid categories like glycerophospholipids or just glycerolipids, we add a little more information, so the predictive power of the model increases slightly. When we then add information of lipid classes such as triglycerides or phosphatidylcholine, we can increase the predictive power of the model even further. And when we then add molecular information on lipid species and lipid subspecies, we can really boost the performance of the model quite a lot, which basically shows that adding information on the lipid species level but also lipid subspecies level improves the predictive power of the model, and is really essential information in order to get a good prediction of obesity for a human subject in these studies. So meaning there is really value added in obtaining molecular information on the lipid composition in order to predict obesity or any other disease in general. So in conclusion, body fat percentage and waist-hip ratio are much better predicted by our model than body mass index, and the error for body fat percentage is only 8%, which is really good. The lipid species detail is needed for accurate modeling, and something I have not shown you, but that is also quite interesting, is that outliers with clinical profiles that predispose for obesity-related diseases also show a lipid profile, based on lipidomics that looks also unhealthy. So that is a quite interesting finding, which would allow us to stratify obesity in the future to get a much better prediction of future health risks than is currently done. And that is something I will come back to in a later slide. So clinical indicators of obesity. So the field methods are pretty okay. Physical measurements are fine, but actually what you need is a metabolic analysis of this, right? You need to look really into the molecular lipid composition. So we also looked at neurological disorders, just to give you a few more short examples on what can be done with lipidomics. Here's a paper on multiple sclerosis published recently. As you probably know, multiple sclerosis is characterized by demyelination of the axons of neurons, and is a very serious disease, including chronic inflammation and neurodegeneration. And this is a typical MRI scan of a patient with multiple sclerosis having, you see this white spots here, which is basically destroyed myelin structures. And we were really wondering whether we can see a lipid signature in the blood for multiple sclerosis. And we did a study with a collaborator from the University of Munich, Michael Simons. And this is the study characteristics. The interesting part here is that it's actually a twin study. So we had a healthy subject and a sick patient, which was twin pairs. So we could really exclude the genetics here from this study. And what we found actually was a significant reduction of ether lipids in the patients. Right, as you can see here, phosphatidylcholine ethers are slightly, but significantly reduced, but also phosphatidyl in ethanolamines. This is just showing you the same analysis on the lipid species level. And what you can see here right away is the prominence of polyunsaturated fatty acid containing lipids. This is just the structure of those lipids. They have, as I just said, polyunsaturated fatty acids, lots of double bonds, and this is basically what these marker lipids for multiple sclerosis have in common. Interestingly, these PUFAs, as they are called, like arachidonic acid or docosahexaenoic acid, are actually precursors for icosanoids, which play a role in inflammation. So the hypothesis that was generated based on the study is that the reduction in polyunsaturated fatty acids is actually the result of the prolonged inflammation in those patients of having multiple sclerosis. So basically, these patients produce lots of icosanoids, which result in inflammation, and this icosanoid production actually consumes polyunsaturated fatty acids. Another example I would like to talk to you about in terms of cohort studies is actually a skin lipidomic cohort study, which is another important area of research that we are also working on. So the skin is basically, as you know, our outermost layer of our body. And there is the layer of the sebum, which is basically the greasy part of the skin, and the stratum corneum, which is basically really the barrier of our body against the outside, which is mainly composed of fatty acids, ceramides, and cholesterol. And this is this part here. And what is known that many different factors actually affect the skin lipidome, like, for example, the skin depth, like depending on where you are in the skin, on which layer the lipid composition changes, the age influences the skin, sex, but also the sampling site. And these in turn, the skin lipid profile then in turn affects hydration of the skin, morphology of the skin, and like, for example, also the barrier function of the skin. And we have developed a methodology that allows us to easily collect skin lipids by using these like small transparent tapes. So we just put a tape on the skin, peel it off, and the lipids stick to the tape. And we can then analyze the lipidome. And what we looked at, for example, was how does the lipidome or the total lipid content of the skin change with age? And what we see here is basically in women, the solid line here, that the lipid content really decreases with age, while for males, it more or less stays robust over a range of many decades, so to say. We can also take a closer look and consider the actual lipid composition, and we see again a clear distinction between the male and the female skin lipidome. We see a strong decrease in triglyceride and diacylglycerol content for the women, while again it stays rather stable for the men. In turn, cholesterol is increasing in women with age, and while, for example, cholesterol esters are staying constant, but also the ceramides are staying constant. So we see mostly a change in triglycerides and diacylglycerols, which are part of the sebum lipid, of the sebum lipidome. So it kind of indicates that with age, the skin of women gets a little drier. So in summary, in skin cohort studies, we have found out that there are age and sex-specific skin lipid compositions. I have not talked about body site specific lipid composition. We have seen that depending on where you take a sample of your skin, maybe made in the face or on the arm or whatever, the skin lipid composition is very different. And also the skin lipid composition in different layers of the skin is very different. And this has clearly implications for the classification of skin types, but also for study design of dermatological cohort studies. So you have to consider all these factors when setting up a dermatological study. That brings me to my next example, which is about basically diabetes type 2. And this is a particularly interesting study because really it allowed us to predict the onset of obesity, of diabetes in the future. And this is a study, again, with our Swedish collaborators. It's a prospective study. So we have collected samples in the 90s, in the early 90s, and followed up on these patients over a period of 23 or so years. And so we knew what kind of disease they would develop in the future. This was a study with more than 4,000 subjects. And like 14% or so of them actually developed type 2 diabetes. And we measured lipidomes at baseline, collected in the 90s, as I said. And the question was, can we identify a lipidomic signature to predict type 2 diabetes? And if so, how does it perform against polygenic risk scores, which are currently widely used in different contexts? And just to remind you what a polygenic risk score is, you all know it's probably single nucleotide polymorphisms, which are point mutations in our DNA and our genome. And each of this point mutation, or many of these point mutations, can be associated with a certain disease risk, right? And taking all those different point mutations together, you can calculate a polygenic risk score. And that tells you something about your genetic predisposition to get a certain disease in the future. So we can do a very similar thing with lipidomics data. I'm not going into detail, it's just an illustration of the complexity of this approach. But when we calculate a lipidomic risk score based on our measurements, we get the following results. So what we have done here in this plot is basically have sorted the subjects for which we have calculated the lipidomic risk score according to risk score. And that results in those people having the highest risk score, the top 10% in this part of the plot. And what we see here in the top 10%, that people with a high lipidomic risk score have a case rate for diabetes type 2 of almost 40%. While based on the polygenic risk score, we have a case rate of something like 25 or so percent, while the null model is giving us something like 14% as was expected based on the cohort composition. So basically what we can see here is that we can identify a prognostic lipidomic signature for type 2 diabetes, and that this lipidomic risk score is working actually much better than the polygenic risk score, which is kind of not so surprising because the lipidomics data actually reflect our current status of our metabolism. So there, a phenotypic readout is much closer to our conditions at the present than a polygenic risk score, which is just describing our predisposition to getting a certain disease. Okay, so coming to my last example on cardiovascular disease, I think we still have a bit of time. I will rush through cardiovascular disease. So again, with our Finnish collaborators, we try to understand how the lipidome is reflecting cardiovascular diseases. But we, in this case, not only looked at the lipids, but also combined that with a genomics approach. And why did we decide for cardiovascular disease? Yeah, as you know, it's a really, really serious and widespread health issue. And it's the leading cause of deaths globally. Yeah, most people will die of heart disease or stroke. That's a fact. And that's why it's important to understand the disease, the biology of the disease or the underlying molecular mechanisms much better. And in this study, we basically try to understand how we can, or we wanted to combine genomic information and lipidomic information in order to get a better understanding of what matters when it comes to cardiovascular diseases like hypertension, stroke, heart failure, and these diseases. And that was published a few years ago already. And again, what was done here, people like our collaborators in Finland looked at those single nucleotide polymorphisms in order to understand like the genetic component of cardiovascular disease, and we looked at the lipidomic part. A nice side result of this study was combining genomics and lipidomics data. It allowed us to actually calculate heritability scores for different lipids. That means we could calculate to what extent the concentration of a given lipid is determined by our genes, in contrast to environmental influences. And an interesting result was that polyunsaturated fatty acids, such as arachidonic acid, for example, have a pretty high heritability score, meaning their composition or their concentration is much more dependent on our genes than, like, for example, like all the other fatty acids in our body. Yeah. Another result of the study was that we actually could identify genetic loci that are significantly associated with lipid concentrations in the blood. And here's just an example for a particular sphingomyelin molecule, which was significantly associated with this particular SNP here. And then we can do for all kinds of different associations. And with that combination of genomics and lipidomics, we could, in this case, actually identify that this particular sphingomyelin is associated with a cerebrovascular disease, which is a particular type of stroke, so-called mini stroke, and might like give clues in the future or be a starting point for future studies trying to elucidate the disease mechanism, or what's like basically understand what is going on in this disease on a mechanistic level. We did this not only for this particular example, we did this for many other different SNPs and types of cardiovascular diseases and lipids. And this study is a really rich resource on the genetic and lipidomic components of cardiovascular diseases. And if you're interested in these aspects of these diseases, then I would encourage you to look into these studies. Okay, so this was my last example, how to use lipidomics in population health studies. And I would quickly summarize and conclude what I have shown you today. So why should population health studies use lipidomics? First of all, it's a very solid technology. It has excellent technical performance, so it can give you robust, high-quality results, which is the basis for everything, right? You require only low sample amounts, and the technology is a high throughput technology, so results for even thousands of samples can be provided within weeks. And the lipidome itself is stable in samples that are stored for years. I haven't shown this, but we have data basically showing that even legacy samples sitting in the freezer for many years, even decades, are robust and stable and can be used still for analysis. And various different study designs like twin studies, intervention studies with a crossover design, cross-sectional studies, or longitudinal studies are possible and can give you really nice data and solid results. And it might in the future, I think we aren't really there yet, but lots of indications are out there that plasma lipidomics, blood lipidomics in general, will enable disease stratification and risk assessment based on molecular and lipid information that goes way beyond the classical clinical chemistry and can really provide added value to disease diagnosis. Yeah, with that, I'm done. I would like to thank you for your attention.